Good day, my name is Karina Roodt from the Soil Health Support Center, which forms part of NemLab. Welcome to today's presentation about testing the health of your soil. Before I get to testing, I would like to give you a bit of information to put the testing into context. We always start with the question, what is soil health? Once we have our context, we look at what we can learn from the tests that are available at the Soil Health Support Center. When we talk about soil health, we take the chemical, the physical, and the biological components into account. Your chemical component is your nutrients, your NPK, the calcium, and other elements that we're familiar with. Your physical component is your soil type, the texture, the pore size, and aggregation. The biological component is the plants, the macro and microorganisms in your soil, which form part of the organic carbon, and the pests and diseases. The aim is to promote the interaction between these components for optimal functioning of your ecosystem. When these components are in balance, you have effective nutrient cycling and the added benefit of microbial action that's physically changing the structure of your soil. This balance promotes infiltration, recycling and retention of nutrients, suppression of weeds, and sequestration of carbon. With testing for soil health, we move our focus to the biological component. All organisms that you find in your soil has a function. They play a specific role in the ecosystem, and these ecosystem services can be used to improve the health of your soil and benefit your crop. For these organisms to flourish, they need a stable habitat with enough resources to support growth and reproduction. To activate as many ecosystem services as possible, interactions between organisms in your ecosystem must be promoted in the soil food web. These interactions have benefits for the soil and the organisms that live in it. One of the key benefits of these processes is nutrient cycling. To us, the core of soil health is the cycling of nutrients, which is dependent on all the components of the soil interacting with each other in a certain balance. We can get carbon and nitrogen free from the air if the biology is functioning optimally, and 78% of air consisting of nitrogen gas is a big potential source of nitrogen. And soils can access this nitrogen if the nitrogen-fixing bacteria are present. But to get the nitrogen to cycle through your soils, you need to get your plants involved as well. Plants use sunlight, carbon dioxide, and water to produce its own food in the form of sugars through the process called photosynthesis. These sugars are used by the plant for its own life processes, and some of it is released into the soil as root exudates. You have bacteria, fungi, and nematodes in the soil that feed on these root exudates. The microbes that have consumed the exudates are consumed by other organisms in the soil food web, and products of decomposition and excretions from these organisms contain nutrients in forms that can be taken up by the plant again. With every exchange, the nutrients are taken up in one form and excreted in a different form. The cycling of nutrients is taking place at the same time, and it's happening all the time if the three components of your soil are in balance. If the biological component is neglected, nutrient cycling stops, the soil food web starts to deteriorate, and you lose the benefits that came with having a diverse soil biota. There are complex interactions, and to test for everything is not practical, but with the right approach to testing, better decisions can be made to make sure that you are building your soil. You have to ask the right question before deciding what test to do, and test results should always be considered in context. The test that we use will answer the following questions. Is there life in your soil? Does your soil show the benefit of microbial activity? Is there food for the microbes? Is there food for the plants? Which microbes live in your soil? And does your soil food web allow for effective nutrient cycling? The Haney analysis combines the results of different tests to answer these questions. You have the respiration test, which measures carbon dioxide that's produced by the microbes in your sample in 24 hours. There's the volumetric aggregate stability test that measures the aggregation. The water extraction test measures the water-soluble nutrients. And you have the H3A extraction reaction which mimics the nutrient exchange in the root zone. H3A is not a chemical formula, it's short for the names of the men who developed this test. To determine the microbial community profile, there are several tests available in the industry. We make use of the PLFA, which tells us more about the fungal, bacterial, and protozoan communities, and the nematode bioindicator test gives us feedback on the soil food web structure and nutrient availability. We will look at the Haney test and see what we can learn from this information that we gather. This is an example of what the test looks like that you will receive from us. This is an extensive look at the nutrient pools, some biological aspects, and the volumetric aggregate stability results. This is what the PLFA looks like. You will also receive a summary of nutrients and mineral composition of your soil and a few important ratios. So let's have a look at how this data is collected. The first question that we want to answer when it comes to soil health testing is there life in your soil? The test that we use to answer this question is the respiration test, also known as the 24-hour burst test. A known amount of soil is dried and then placed in a known amount of water to simulate a rainfall or irrigation event. This triggers the microbes in the sample to respire and produce carbon dioxide, which is measured after 24 hours. The amount of carbon dioxide that is measured is used to calculate the microbial biomass, which is used to estimate the potential mineralizable nitrogen that can be made available by these microbes during a growing season. This number also correlates very well with the food that's available for microbes and with soil organic matter numbers. It's a very straightforward test in that a higher number indicates more fertile soil. This test is more focused on the bacterial populations because they are big role players in converting organic nitrogen into inorganic nitrogen that can be used by the plant. Degraded soils will give a reading below 30 parts per million. Agricultural soils where soil health principles have been applied for a few seasons will give readings between 70 and 120, and we get soils that measure far over 300. And different soil types give different results, so it's important to know what to expect and to take baseline samples to know how you are improving. The next question that we want to answer is, does your soil show the benefit of microbial activity? The test that answers this question is the VAS test, or the volumetric aggregate stability test. In very basic terms, the test measures how much aggregates are accumulated around the soil particle. The aggregates is a combination of organic matter, debris, minerals, dead plants, and microbial materials, and very importantly, it contains molecules that holds these components in place, and this aggregation can only be achieved over time with extensive microbial activity. The soil is dipped in water so that only stable aggregates remain. The stable aggregates are mechanically removed, and the difference in diameter is used to calculate the volumetric aggregate stability percentage. This is a great way to see how the soil has been changed through microbial action. The aggregation is an important indication of the soil's ability to resist erosion. A percentage below 30% is considered low. Agricultural soils would typically be between 30 and 45, and anything above 45 is considered high. Sandy soils will always test low, but it is still worth monitoring this number. As we move on, the answers become more complex, so let's take a look at how we measure before we look at what the numbers mean. We use different extraction techniques to find out about the different nutrients in the soil. Water is used to extract the soluble organic carbon and the total available nitrogen. The H3A extractant is a combination of weak organic acids that mimic the action around the roots of plants, which causes the release of nutrients that can be taken up by the plant. From the data that we collected, we can now answer the next question. Is there food for the microbes? We look at water extractable organic carbon, which is measured, the water extractable organic nitrogen, which is calculated, and the ratio of these two components. The water extractable organic carbon is simply the water-soluble carbon measured in your sample, and this is the carbon that's used by the microbes as a source of energy. These carbon pools are replenished by photosynthetic products released into the soil through the roots of the plants, i.e. your cover crops. The threshold for organic carbon is a rough guideline, since the important part is really the C to N ratio, but for water extractable organic carbon, numbers below 170 is low and above 350 is considered good. The water extractable organic nitrogen is calculated by subtracting the H3A extracted inorganic nitrogen from the total water extractable nitrogen. If your soil food web is functioning well, this nitrogen pool can be replenished by adding legumes to the system. Again, it's more important that your C to N ratio is correct, but you are aiming to get water extractable organic nitrogen numbers above 10 and above 20 is very good. When you have these numbers, you look at the carbon to nitrogen ratio. The microbes consume the organic carbon and organic nitrogen together as a source of energy. As the microbes use the carbon and nitrogen, they convert the organic nutrients into inorganic nutrients, which can be used by the plant. This is a multi-step process that involves different organisms at different levels in the soil food web. The carbon to nitrogen ratio answers the questions of food availability for microbes with a focus on releasing nutrients to be used by the plant. A carbon to nitrogen ratio above 20 to 1 generally indicates too high carbon for the amount of nitrogen that's available. The chemical process of burning the nutrient as a source of energy will not happen, and no nitrogen and phosphorus will be released to the plant. As carbon inputs are slowed and legumes added to the system to increase the available organic nitrogen, the carbon to nitrogen ratio will decrease. Now the organic carbon and nitrogen can be used as a source of energy, and the nitrogen and phosphorus is made available to the plant in inorganic forms. When the carbon to nitrogen ratio drops too low, it means that the organic carbon is too low and the organic nitrogen is too high. Carbon inputs must be increased. You do this by incorporating living roots of photosynthesizing plants. If your carbon to nitrogen ratio drops too low, the microbes can start to use the stable carbon as a source of energy. And this means that they are not building your soils, that they are actually burning through your soil organic matter, the carbon in your soil organic matter. So it's really important to keep that organic carbon number high enough in ratio with your organic nitrogen. The ideal carbon to nitrogen ratio is between 10 to 1 and 12 to 1. When your system has reached this point, you have to attempt to keep increasing these nutrients in this ratio so that your microbes can continue to flourish. These nutrient pools are replenished by the interactions in the soil when the soil food web is functioning optimally. Ensuring that there is a source of energy for the microbes is the first step in creating a habitat for them to flourish and for your soil to benefit from these life processes. All this information is summarized in the Haney number or the soil health score, which is calculated using soil respiration, water extractable organic carbon, and the water extractable organic nitrogen numbers. Above 7 is considered a good score, and it is suggested that you take a sample from the natural veld in the same area to compare with a sample taken from the agricultural soils, just so that you can get an idea of what the soil's potential is. When there's food for the microbes, food and nutrients will be made available for the plants. So this brings us to the next question. What nutrients are available to the plant? The Haney test gives the total nitrogen, phosphorus, and potassium and other nutrient factors. The nitrogen and phosphorus amounts are further divided into organic release, organic reserve, and inorganic totals. And if your microbial activity is low, your system will show reserve amounts. This means that there are organic nutrients available to the microbes, but there are not enough microbes to do the work of breaking down these nutrients. When your farming practices have allowed the microbial populations to increase, the amount of reserve nutrients will decrease until it becomes zero. And that just means that nutrients that are available to the microbes can be utilized by the microbes because there's enough of them to eat the food that's available to them. For a quick reference, the organic nitrogen to inorganic nitrogen ratios are also included in this report. Above 5 is ideal, and it shows that most of the nitrogen is in the organic form, and the nitrogen will be made available to the plant through microbial activity. If this ratio is below 5, it usually indicates the use of fertilizers. And the Haney will help to lower your synthetic inputs by giving you a clear picture of what the organisms in your soil can contribute. So far, we looked at the nutrient availability, which is both influenced by and dependent on microbial community. The first test that we look at to give us insight about the microbial community profile is the PLFA test. PLFA is short for phospholipid fatty acid. The phospholipid fatty acids are molecules that consist of a glycerol molecule attached to a phosphate on the one end and two fatty acids on the other end. These molecules interact in a very unique way to form the phospholipid bilayer. The phospholipid bilayer is the membranes of cells of living organisms, even you and I. It's the casing that helps cells to keep its inside on the inside and the outside on the outside. Different organisms have different size phospholipids, and these variations in sizes is used to analyze the microbial community composition with regards to bacterial, fungal, and protozoan communities. When you look at the PLFA result, you should be less concerned with the numbers and rather look at the result as a feedback on the habitat that you've created in your soil. Changes to your farming practices will influence the soil biota, and it will be reflected in this result. The most important part is the microbial biomass and the diversity index, so you look at that first. If you can get your microbial biomass over 3,000 and your diversity index over 1.4, then you're heading in the right direction. You'll be able to see your nitrogen fixing bacteria when legumes were planted. You'll be able to get an idea of the presence of mycorrhizal fungi, so you know that your crop has microbial support in taking up phosphates. The fungal to bacterial ratio is related to physical soil disturbance, and your predator-prey ratio will indicate the possibility of nutrient cycling through the trophic levels. And this brings us to our next question. Does the soil food web allow effective nutrient cycling? Here we look at NemLab's nematode bioindicator test. It's also known as the quadrant test or the Colonizer test. This test is a direct identification of nematode groups, and this relates to other organisms in the soil as well as the habitat where they live. So what are nematodes? Nematodes are a diverse group of organisms that can be found in many different environments. They are unsegmented, they can be microscopic to several millimeters in size, they are bilaterally symmetrical, thread-like, and some of the females have swollen bodies. Nematodes can fulfill many different functions in the soil, and their excretions can add up to 30% of the total mineralizable nitrogen in the soil. They have a role in sequestering carbon, and they help improve soil structure. They can regulate disease-causing microbes, but they can also be vectors for diseases, or they can cause direct crop losses. There are many species of nematodes that belong to different feeding groups, and this is one of the reasons they are seen as an excellent indicator of soil health. We use nematodes because they fulfill the following conditions as an ideal soil microbiological and biochemical indicator of soil health. They are simple to measure, and we have sufficient taxonomic knowledge to identify organisms accurately and efficiently. It works equally well in all environments, and it's applicable to all geographical locations, soil types, and vegetation types, and they reliably reveal which problems exist, whereby reflecting the structure and the function of ecological processes, and they respond to changes in soil conditions that results from land management practices. The core of the Colonizer test is that the more of a specific food there is, the more of the nematode that feeds on that food there will be. For instance, if you have high amounts of bacteria, you will see increased levels of bacterial feeders. If you have more fungal system, you will have more fungal feeders. And looking at the soil food web, we can see it consists of many trophic levels that are interconnected, and the nematodes can be found on most of these levels. The nematode bioindicator test gives a layout of the diversity of nematodes, which will reflect the biodiversity of the organisms that they feed on. The indices are calculated to quantify the state of the soil food web. The Colonizer Persister Scale forms the basis for these indices. Nematodes differ in their life strategies, which can also be used to get information about the habitat where they are found. The Colonizer Persister Scale ranges from early colonizers of extreme enrichment to general opportunists of new resources to persisters in undisturbed habitats. Resource availability, so the amount of food available to microbes, can be derived from the colonizer nematodes in the population. The soil food web complexity and connection connections as well as the stability of the system is revealed by the persister nematodes. Looking at the indices with this information in mind, the structure index will indicate the state of the soil food web as influenced by stress and disturbance. You're aiming for 100%, but most annual crops won't go higher than 50% due to regular disturbance. The enrichment index is a measure of the resources that are available as well as the level of decomposition. The value should fluctuate around 50%, and high values simply indicate that the nutrients have not been decomposed yet. The channel index indicates the predominant decomposition pathway. A high value indicates more fungal decomposers, so it means that the food resources that are available are high in lignin and cellulose. A low value indicates more bacterial decomposers, which indicates the availability of more nitrogen-rich resources. Remember, the more of a certain food there is, the more of the microbe that feeds on it, the more of the nematode that feeds on that microbe. The basal index indicates if the bacterial system can provide fast nutrient turnover. More fungal systems have slower nutrient turnover. The fungivore-bacteriivore index is determined by the presence of nematode groups that are associated with stressed environments, and a higher number is expected for fallow soils and a lower number for soils that have cover crops. The enrichment index and the structure index is used to plot a point on a matrix. You want your soils to fluctuate around 50% enrichment and get as close to 100% for the soil food web structure. When you do this test year after year and you apply the soil health principles, you will see that point that's plotted moving from left to right towards the 100% and fluctuating around the 50% enrichment. Soils from conventional farming systems is expected to show depleted resources and minimal soil food web structure. These soils will be vulnerable to pests and diseases, and the only way to improve from this point is to apply the 5 soil health principles. Cover the soil, whether with cover crops, mulch, or compost to trap moisture and to keep the soil cool. Living roots help water infiltration and it will feed the microbes. Diversity above ground creates diversity below ground. The less soils are disturbed, the more structured the soil food web will become, and if possible, incorporate an animal component. Enriched soils that is highly disturbed will only improve if disturbance is decreased. Remember, this does not only refer to physical disturbance through tillage, but also chemical disturbance. Undisturbed soils have more diverse communities of microbes, and they need their energy resources replenished regularly. So keep feeding the microbes to keep your enrichment in the area of 50%. When the enrichment is high and the soil food web structure is high, the microbes must be given time to decompose those available nutrients. So to summarize, if you want to test for the health of your soil, it takes an integrated approach. You have to think about what you want to learn from these tests, and you can use these questions to guide your decision. Is there life in your soil? Does your soil show the benefit of microbial activity? Is there food for the microbes? Is there food for the plants? Which microbes live in your soil? Does your soil food web allow effective nutrient cycling? The tests that are done locally at our lab in Klapmuts is the nematode community profile, the mycorrhiza colonization test, which we haven't discussed today, the respiration test, and the volumetric aggregate stability percentage. Most of the Haney is done at Ward Laboratories or Region Ag Lab in the USA, along with the PLFAs. I'd just like to take this opportunity to thank Sheila for her dedicated mentorship and all the resources to put together this presentation. I would also like to thank the NemLab team who works tirelessly in the passionate pursuit of great service and healthier soils. We hope that you've enjoyed this presentation. Feel free to email us with any questions.